We could have scrapbooks, I'm sure, for all the pictures we've taken over the years in our lab. So thank you all for being here. Sorry we're a little delayed. I wanted to make sure everybody knew my defense was starting. I'm just kidding, I did not pull the fire alarm. So my name is Chelsea Bonnain, and today I will be telling you about the work I completed for my master's thesis entitled Iron-Virus Interactions, the Development and Testing of the Ferrojan Horse Hypothesis. Oh, one quick thing, if you tweet, we're all about science communication. If you tweet for science, it's my Twitter handle, and I made myself a hashtag that's DefenseIronPhage. So feel free to tweet me questions or live tweet if you feel like it. All right, so just a little overview of what I'm going to cover today. Like Maya said, my work has been very interdisciplinary, so I want to give you a little bit of background on the two disciplines, marine microbiology and iron biogeochemistry. And the Ferrojan horse hypothesis was this really cool idea we developed, so I'm going to cover that today. And then I'm going to tell you about the methods we developed to test the Ferrojan horse hypothesis using a model system. Lastly, I'm going to give you some conclusions and where this work is headed moving forward. So to start, iron is essential for multiple domains of life. We as humans require iron to oxygenate our blood. Within our gut, pathogenic bacteria such as E. coli have an iron requirement as well. And even in the oceans, phytoplankton, including both eukaryotes and cyanobacteria, have an iron requirement to grow. Now, iron makes an excellent cofactor for a variety of enzymes and proteins, which is why it's used in multiple cellular processes. To name a few, although there are an abundance of them, iron is a cofactor for a DNA repair enzyme, so it is required for DNA synthesis. Iron is also bound to a ferrodoxin protein, which is involved in electron transport, nitrogen assimilation, and respiration. Iron also is used as a precursor, bound to a precursor protein that is involved in chlorophyll production, so it is required for photosynthesis. And iron is a cofactor for an enzyme that is responsible for the removal of reactive oxygen species in cells. So if iron's so important, it shouldn't be a problem to get it, right? Well, for these phytoplankton, it's actually a pretty big challenge because a majority of the iron in the ocean is actually insoluble. So as dust lands on the surface of seawater, at the pH of seawater, it actually forms a precipitate and sinks out of the upper water column away from where phytoplankton can access it. The remaining iron is going to be mostly dissolved. So this is a map of the nutrient limitation for small phytoplankton. I got a little ahead of myself, sorry. So you can see by the purple, the majority of the surface ocean is limited by nitrogen or nitrate. There are regions of the ocean where there's excess nitrate, so there could be more primary productivity in those areas. A couple examples include the subarctic Pacific, the equatorial Pacific, and the southern ocean around Antarctica. In these regions, you can see the blue, and that's because they're largely limited by iron. We call these HNLC, or high nutrient, lower than expected chlorophyll regions, and they actually constitute about 40% of the global surface ocean. So why isn't there any iron? Well, it's insoluble at the pH of seawater. So the majority of that iron will precipitate out. What's remaining is sub-nanomolar concentrations of dissolved iron. And of that dissolved iron, more than 99% is actually complex to some form of organic ligand. So here's sort of a spread of the different iron pools. Within the green, we have the particulate iron. That can include bacteria, any large biogenic iron. As we move below 0.2 microns, we get into the dissolved iron fraction, and that's made up of both the colloidal and the soluble fraction. The colloidal fraction is between 0.02 and 0.2 microns, and does consist of a lot of iron-binding ligands. However, besides some humic substances and exopolysaccharides, transparent exopolymers, we don't know what a lot of these iron-binding ligands are. Below 0.02, we have the soluble fraction, and one of the most well-studied iron-binding ligands within the soluble fraction are siderophores. Siderophores are small molecules that are produced by bacteria and fungi to find iron and bind it with a really strong affinity. These are some of the strongest iron-binding ligands we know, and then can bring that iron bound to the siderophore back to the cell. They can either reduce it at the cell surface or uptake it to a siderophore receptor, through a siderophore receptor. Some marine bacteria also produce these lipid chains to tether the siderophore iron complex because things are moving around in the ocean. They don't want to get them too far away. So there are also iron transporters that can take up or reduce this iron at the surface, and that's where our iron ligands would come into play. But like I said, we don't know what a lot of these are. So when we want to look at these dissolved iron pools, our first step, and we often do the shipboard, is to physically partition that seawater sample. So what that looks like in the lab is we take a whole seawater sample with all of our iron. We're going to put it through a 0.2 micron filter. What goes through that filter is the dissolved. What is excluded is the particulate iron. We then take a subsample of that dissolved iron, put it through a 0.02 micron filter. What goes through is the soluble. We can calculate the difference between these two iron fractions to give us the colloidal iron. Now, that's all well and good from a chemist perspective. As a biologist, we know it's a little bit, there's a lot more here. So what else could possibly be in this seawater sample? We have bacteria, like I mentioned, and siderophores, just to name a few of these sources of iron. Well, the bacteria we're going to go ahead and we're going to filter out. So it's going to get caught in a 0.2 micron filter. It's not part of the dissolved, it's part of the particulate. The siderophores, we're going to go all the way through the 0.02 into the soluble. And like I mentioned, a lot of this colloidal iron, we don't know what these ligands are, although some studies have calculated they constitute up to 80% of the dissolved fraction. Siderophores, although very well studied and very strong iron binding ligands, in some studies of the Northern Atlantic calculated they constitute only 0.2 to 4.6 of the soluble fraction. So we're missing a lot of different iron binding ligands within this colloidal fraction. So what else could be there? Well, as a virologist, we use these filters a little bit differently. The way we do that is we filter out our bacteria, but we try to get our viruses. And what happens is they go through the 0.2 micron filter and they get caught on a 0.02 micron filter. So they are actually, while not truly dissolved, part of the colloidal fraction of iron. But what would viruses even matter? I mean, what difference could they possibly make? Well, in case you didn't know, viruses are very abundant. There are viruses for every organism in the ocean. So there are 10 million viruses in a single milliliter of seawater. Here's a picture of water that I just scooped up from Bayboro Harbor right outside. And I put it through a 0.02 micron filter. I stained it with a nucleic acid stain, and I looked at it under an epifluorescent microscope. So as you can see, these big bright circles, those are all bacterial cells. Those little pinpricks everywhere, those are viruses. So they're out there. So viruses are the most abundant biological entity in the oceans. The second most abundant being bacteria. So the majority of these viruses are actually going to infect bacteria. These are called phage, short for bacteriophage, or a virus that infects a bacterial host. So your sort of iconic image of a phage is pictured here. The capsid is actually a protein structure that surrounds the nucleic acid inside. This could be DNA or RNA. This central tail sheath is like a tube. So when the phage is infecting its host, it can push that DNA or RNA through the tube into the host, and that's the only part that enters, none of the protein. These protein structures are long tail fibers, the base plate and short tail fibers, and those all help the phage attach to the host surface and absorb for infection. So what that looks like for a lytic infection, if you look at the top here for the attachment stage, those tail fibers, the base plate and the short tail fibers, all help that phage find a very specific receptor. So if you think about in a liquid or a fluid, these phage are kind of bouncing around and hitting things, and they need something to attach to or they're just going to fly off. They can't really infect. So they find their specific receptor on the host surface, they attach, they sort of launch down into the cell surface, and they can inject their nucleic acid into the cell. That phage genome will then take over host cellular mechanisms and tell the cell, make a bunch of phage. It produces all the different proteins and nucleic acids to make the new phage progeny. Once they are fully assembled and the burst size becomes critical, the bacterial cell lyses open and releases all the new progeny phage along with a ton of other nutrients. The reason that's important is phage have been characterized, or viruses as a whole, have been characterized for their contribution to nutrient cycling. Every lytic event of a host is going to release a ton of nutrients in the upper water column that can be recycled. This process is called the microbial loop. So this is a little bit complex, but all I want you to take away from this is that there is consumption and production of dissolved organic matter from microbes throughout the upper water column. This dissolved organic matter can be recycled or remineralized by bacterial heterotrophs. Viruses, or the viral shunt, sits at the middle of this microbial loop, and what it does is it lyses open bacterial cells and enhances the productivity of that microbial loop by feeding more and more dissolved organic matter that can be remineralized. So there's more and more recycling. Alternatively, there have been studies of what's called viral shunt, which is where that bacterial lysis has a bunch of sticky parts that can actually clump together and form particulate organic matter that is actually heavy enough to sink out of the upper ocean and into the deep ocean. So the phage particles themselves, or viruses, have also been characterized for their contribution to macronutrients, carbon, nitrogen, phosphorus. So that protein structure, that DNA, those all contain these elements. So that's actually been quantified and well studied, but the trace metal contribution has not yet been investigated to our knowledge. So this is kind of an exciting field to look into farther. Promised I'd drink water in between. Okay. So that brings me to the Ferrojan horse hypothesis. And when we started looking into this, I was literally looking through all these papers for iron and viruses, iron and phage, any sort of connection between the two. And what I found was actually from medical literature. So the viral research in the oceans is actually kind of behind that of the medical field, because we study bacterial model systems to look at different proteins that are homologous to humans. So for a long time, we've been looking at these different proteins in bacteria, trying to develop antibiotics. So we know a lot more about bacterial host systems for pathogenic bacteria than we do for the oceans, and their phage we know a lot more about as well. So I actually found this paper. It's a study by Marc van Raja's group in Spain. They did something called X-ray crystallography, which is just a long name for solving a protein structure in incredible detail. So this is, again, my picture of my phage. This is actually T4, kind of our iconic phage. It's arguably one of the most well-studied phage in the world. It infects E. coli. So there is this tail fiber here, and there is a tail tip protein called GP37. And when they solved the structure of this GP37 tail tip protein, what they found were 7 iron atoms. These were coordinated by paired histidine motifs. And there were 7 of these histidine motifs, which they said for each histidine motif, it probably corresponds to an iron atom. So these 7 iron atoms were described. This was modeled for us by Dr. Leon Hardy at USF St. Pete. So we wanted to see if these histidine motifs were anywhere else, because we don't have these protein structures for a lot of different phage proteins for pathogenic bacteria, let alone within the ocean, for pathogenic phage, let alone in the ocean. So we looked into these histidine motifs by the sequences of different phage proteins. And of course, here's our E. coli phage T4, beautiful image by Tony Greco, somewhere out there, I think. Thank you. And so we know those 7 histidine motifs correspond to 7 iron atoms. We found in phage lambda, one of the tail fiber proteins, also had 7 histidine motifs, so we predict maybe that corresponds to 7 iron atoms as well. Within the oceans, a Prochlorococcus phage was found to contain 6 of these histidine motifs, so perhaps that's 6 atoms. And then a couple Synechococcus phage, we found 4 and 11, so maybe there are really variable amounts of iron atoms in these phage. They all have different numbers of tail fibers sometimes, so that could change sort of this variable of how many iron atoms we're finding in these phages, if it's there at all. And then from the Mediterranean, there was a phage that was isolated that we don't know the host, but we also found 7 of these histidine motifs. So this is really exciting. This was sort of like, okay, this could be happening. And it's in E. coli, but that's a model system that, is definitely we can work with, and we can see if these are conserved throughout more of the ocean's databases for phage by doing some bioinformatics. So we looked a little deeper, and we found these papers from like the 1970s. And what they were studying was a siderophore receptor on the surface of E. coli. And I'm pretty sure they were trying to figure this out, solve the protein structure so that they could develop antibiotics. It seems like a good strategy. If a siderophore receptor is essential to a bacterial cell to get iron, then it's not going to get rid of it. So if you dissolve an antibiotic, design an antibiotic that's an antimicrobial agent attached to siderophore, it's a good way to target a bacterial cell. Well, phage thinks so too, because apparently they described that when they knocked out specific proteins in that siderophore receptor complex, they actually inhibited the phage from infecting. So they described this as a competition between the phage they were working with and a siderophore bound to iron for the siderophore receptor. They actually found by manipulating this protein, they inhibited multiple different phage from infecting the E. coli cell. And so we have a little bit of a battle going on. So this is all very exciting, and we put all this evidence together from the model systems to create the Ferrojan horse hypothesis. I'm going to walk you through this. So at the top here, we first have a phage encountering its E. coli host, and it has to gain access to that receptor. Now, bacterial cells are constantly evolving to not be infected by phage, and phage are constantly evolving to infect them. So if these phage are using iron in their tail fibers as sort of a Trojan horse, or a Ferrojan horse, if you will, it could gain access to that siderophore receptor because it's disguising itself as that precious iron, and then it can attach to the cell surface and inject its nucleic acid and then take over the host cellular mechanisms. At this point, the phage genome would tell the bacterial cell to make a bunch more phage proteins and nucleic acid. It would assemble those phage proteins into the new progeny. We predict at this stage, the Ferrojan horse hypothesis states that perhaps the iron from within the bacterial host storages would be incorporated into new phage progeny. This seems likely if you think of like a marine system where there's no iron in the environment and then there's a bunch of iron in these bacterial cells. It seems like if you're phage, you're taking everything you can, getting out, bacterial cells done. So why not take the iron too? But it just hasn't been shown yet. So the last step is the lytic event, the lysis of that bacterial cell. Releases the new phage progeny, but it also releases all those nutrients I told you about, including the trace metals. While these systems and the contribution of trace metals by viral lysis has been well described, however, that iron could in fact be complex to new progeny phage, and then it might not be available for remineralization. So that could be an important component to this iron cycling that hasn't yet been considered. So to conclude a little bit about the Ferrojan horse hypothesis, the three corollaries that we stated were that dissolved iron could be complex within phage, and this could be part of the colloidal fraction of dissolved iron. Phage could use iron as a Trojan horse to gain access to that siderophore receptor for infection. Lastly, cellular iron could be recycled from host storages, put into phage proteins, and therefore unavailable for remineralization. So some insight we gained from the Ferrojan horse hypothesis, we did sort of these back of the envelope calculations to try to figure out best worst case scenario, and we calculated for your average dissolved iron concentration, about 25% of that being colloidal on an average for the surface ocean, the global open water surface ocean, and for that the 10 million viruses in a milliliter of seawater, assuming that they're all phage, which is an over-assumption, and that there would be those 42 iron atoms per phage, and I got that by doing the 7 iron atoms, assuming they all have 6 tail fibers. So a lot of assumptions, but we wanted to see sort of what could viruses even contribute to the colloidal fraction. Well, we got up to 70%. of that colloidal fraction could be actually bound to phages. And this could be variable based on all those assumptions I just told you, but it kind of gave us a starting point and something that we could test. Next, we predicted that based on your average iron content within bacterial cells, your average burst size of viruses, that 42 iron atoms per phage, that there could be up to 14% of the iron that would have been released to be recycled for remineralization could actually be incorporated into progeny phage. One thing I didn't mention is that siderophores, you know, we really care about. You know, there are these very strong iron-binding ligands, but they constitute sometimes 0.2 to 4.6% of the soluble fraction. So even if this number isn't 70% for the phage contribution to iron, any amount of iron that is actually a part of the phage structure even could contribute and have implications for the way we think of iron cycling. So as much as I just want to like dive in to the water and go for iron and phage, it's really not that simple. There are a lot of challenges that we have to overcome. I would start with a marine model system, but marine phage are very diverse. We only have about 1% of the diversity of bacterial cells, so we really don't have all those different marine phage in culture. And that can be a challenge. So the marine phage proteins, of course, if we need the phage to study the proteins, we don't always know, we don't have that structural evidence, that hard evidence that there's iron in phage proteins, especially for the oceans. And a lot of these host receptors are just unknown. So, you know, it's great to work with E. coli in a model system because those antibiotics were being developed, but it's not so easy to apply that to the marine environment. So it was really necessary that we start with sort of a proof of concept to develop these methods using a well-known model system. Okay. So, which leads me to my next part. How I tested these aspects of the Ferrojan horse hypothesis using a model system. My questions I wanted to answer were how much iron is incorporated into progeny phages. I just threw that number 42 at you based on that tail fiber structure that we know contains iron for a pathogenic phage. However, we don't know if there's any other protein structures that could be binding iron, or there are various phage throughout the oceans that don't have tail fibers. So their capsids can be kind of sticky, maybe they're sticking iron. Some of them have less tail fibers or no tail fibers. So there's a lot of variability. Although those proteins can be conserved, there's a lot of different things that could change that 42 number. So my next question is, does iron in progeny phages originate from within the bacterial host cell? Like I said, it seems like it could be likely, but it just hasn't been shown yet. So to test some of these using phage that we know and love very well, we used E. coli, and it's sort of our lab rat of the microbial world. And here's a picture on the left of an E. coli cell being assaulted by T4. I appreciate its sacrifice for science. And in the middle, I have my beautiful T4 in culture. Again, thank you, Tony Greco. And as you can see from this image, there is the capsid here with the nucleic acid inside. The tail tube, it's a myoviridae, so it's actually a contractile tail tube. So it will sort of inject the DNA once it gets to the surface of the host cell. And then you can actually even see these tail fibers surrounding the phage, which was very exciting. And so T5 is a Cyphoviridae. So it has a longer, non-contractile, sort of bendy tail and only 3 tail fibers, but it also infects E. coli. So we know T4 has these iron atoms in the tail fibers, so that should be like a positive control. We do not know about T5. We do not have structural evidence for those tail fiber proteins. What we did find are these, not the histidine motifs, but these other iron-binding motifs that bind iron-sulfur clusters. So it kind of was a wild card. We might see an iron signal or we might not. So a little bit about my experimental design. I used iron 57, which has low natural abundance as a tracer for the iron to progeny phage. So what I did was I took bacterial cells, I grew them in media with iron 57 as the sole iron source, And then I allowed them to take up that iron 57 as they were actively dividing and growing. And then I rinsed them a ton of times to get rid of any excess iron that could be in the media, because I want to make sure the only source of this iron 57 to the phage is from in the bacterial host cell. So then I infected with either my phage T4 or T5, allowed them to assemble. At this point, we predict that perhaps the iron 57 from the cell, if that is the source, would be incorporated into the progeny phage. And then upon lysis, I'm able to purify the phage, and I can actually see that iron 57 signal. So I want you to think about this for a minute. Like, here's my E. coli cell, and here my phage. And you're like, why is it holding a bat? Oh, no. Well, think of this bacterial cell for a second. It's like a piñata. So these phage are going to break open that bad boy and release all the goodies inside. Because if you think about this, even for, E. coli in culture is weird, then in the oceans, this is breaking open a bunch of good bits that other bacteria can come and get. So these are those bits. Might have some different proteins, like some flagella and pili, some outer membrane proteins, maybe some other small cellular debris, ribosomes, other bacterial colloids that could be a similar size and density as phage, and of course, our hero, the iron 57 phage. Golden ticket. So I'm going to go through a process of purifying these phage, because I would love to just go pick out my favorite candy and be like, got it, we're good. It's not that simple. So I have to go through some different procedures. The first one being centrifugation, which is basically just spinning these tubes of lysate, or everything that was burst open by the phage, and just spinning them down really fast. And all the debris is going to pellet, not all of it, but the majority of the bacterial debris that's heavy is going to pellet to the bottom. And my phage will remain in what's called a supernatant. So the outer membrane proteins, the big clunky stuff, is going to be removed. I'll then filter that supernatant, that liquid I get, through a 0.2 micron filter. And what goes through will be my phage. And then what's excluded might be some bigger stuff that doesn't get through that pore size. Next, I'm going to do what's called ultracentrifugation. So it's even more extreme than the last one. And that is going to be spinning tubes really, really, really fast for a long time. And it's so strong that it can actually pellet those phage to the bottom of the tube, and everything above what's called a sucrose cushion is excluded. And so I can resuspend that phage at the bottom and help to purify them from other sort of maybe proteins of a different density. And the last step is dialysis, which will remove any other small cellular debris that might still be left over in the sample. So what I'm left with is this purified phage lysate, but how do I know there weren't other bacterial colloids that didn't purify with them? And what if they have iron 57? Well, I have a bacterial control for that. So what's really great about this system is I have all of my different phage in this mixture. If there are any other colloids with iron 57, I can actually take into account those other bacterial colloids by a control that was by phage. It was lysed mechanically with chloroform, and I can get my purified iron 57 phage. Okay. So that was fun, but this is the real deal. I have a cell culture. It's homogeneous. I will then divide it into separate flasks, and I have to go through that rinsing process that I mentioned. What that looks like is centrifuging all of these bacterial cells to pellet them, and in this case, they remain intact, so they're safe. Everything else in that supernatant is going to be my media with any excess iron 57 that wasn't taken up by the cells while they were growing. And I want to get rid of that because I don't want any other source of iron for these phage, right? So I will pour out that supernatant, I'll replace it with a media that has no iron added, and I'll spin it a lot. I want to rinse them off with that clean media, and then I will pellet them again. I do that 3 times. So what I should end up with is a bacterial pellet that is enriched in iron 57 within each bacterial cell, but a supernatant that is really depleted in iron 57 because I don't want any excess that the phage can access. So then I divide these, this was all still divided. These are all in triplicate. I have my T4 infected, my T5 infected, the bacterial control I mentioned lysed by chloroform instead of phage, and then my media blank has no bacteria, no phage. So I lyse these, and then I take my lysate and I need to pellet the debris. So the next step is the centrifugation. So what I end up with is this unfiltered lysate and the bacterial debris, removing all the pelleted to the bottom. I take that unfiltered lysate and I need to filter it for size fractionation. So I put it through a 0.2 micron filter. What goes through contains my phage in the dissolved fraction. I then take a subsample and I put it through the 0.02 micron fraction to get at the soluble iron. And I can take the difference of those two and calculate how much of that iron is colloidal. I then take my dissolved fraction and I put it into the ultracentrifugation, the sucrose cushion, and I spin it really fast, my phage pellet to the bottom, and so any other label that's left in that sample that's a different density than the phage will remain at the top. So I can pour it out, I then resuspend my phage pellet in some buffer, and I dialyze it for the last step. Over multiple different dialysis buffer changes, what I should end up with is my purified phage samples with the iron 57 label. Okay. So in the lab, I've had to, with the help of Salvo, where he is, thank you. I think I horrified him enough times when I tried to use like metal scupulas and glassware and an autoclave. I mean, all those things add iron contamination. So as a microbiologist, it took some craftiness for us to work out these methods in the most clean way we can, because I mentioned that iron 57 has a low natural abundance, but iron 56 has a high natural abundance. So if we get any contamination, iron 56 added to the system by dust or anything metal, or that could be preferentially taken up by the bacterial cells, and then we kind of lose track of our label, or by the phage if they're taking it up from the media. So we really want to keep things as clean as possible, as a trace metal clean as possible as a microbiologist. So within my laminar flow hood here, I have a setup for how I do my size fractionation, so filtering for dissolved and soluble. All of my materials were cleaned in a trace metal clean soap bath. We acid cleaned them for a month, and then we left them to dry and in a trace metal clean hood, and double bagged them so that no dust could fall. They couldn't get contaminated, as best we could, couldn't get contaminated by iron 56. So I take both my original bacterial cells, and I want to quantify how many I have, and then I take my final phage samples that are purified, and I also want to quantify those. So what I do is I stain the sample, filter the sample, stain the filter with Cyber Gold. It's a nucleic acid stain. Here's a picture of the epifluorescent microscope and a beautiful image of all my phage and culture. So I can count those, and then this is a picture of my E. coli cells. And those are also lit up by Cyber, and I can quantify them as well. The beautiful Element XR high-resolution inductively coupled plasma mass spectrometer was so beneficial. I got to do all of my analyses for iron 57, iron 56 on this beautiful instrument. And of course, we want to quantify the iron 57, but I mentioned we want to reduce iron 56 contamination, but if it did happen, we want to know so that we could account for the percentage of iron 57 that is introduced for any iron 56, and we can subtract that out of the sample. Results. So just to orient you a little bit, this experiment was repeated multiple times today. I'm going to tell you about 3 iterations of the experiment all the way through. So in green I have T4, blue I have T5, red is my bacterial control with the lysis by chloroform, no phage, and yellow is my blink. On the y-axis I have the iron 57 content in nanomoles, and along the x-axis I have, starting with the cell culture pre-lysis, so my intact pinata, each of the steps post-lysis to purify those phage. Okay? So pre-lysis, if we look at these values for iron 57 in nanomoles, we see that we get about similar quantities of iron 57 for each of our bacterial cultures. And that's what we want to see. We want to know that starting with a homogeneous culture that has iron 57 provided to these phage. We break open the pinata. It's not going away. And so the first step is to centrifuge. So everything that is in that unfiltered lysate that's still left, we might have removed maybe some large outer membrane proteins. So here we have iron 57. In T4 and T5 you can see stays a little bit higher. And then in bacterial control, it's a little bit more depleted, although they all went down because we're filtering out some of that other stuff. The dissolved fraction is after 0.2 micron filtration. So we're going to get rid of some of that big stuff. And again, you see T4 and T5 stay more enriched while the bacterial control goes down. The colloidal fraction was just taking the difference between the dissolved and soluble. So what's interesting here is the majority of that dissolved fraction looks like it actually was colloidal. And then you can't see these on the scale bar, so let me blow it up for you. Ultracentrifugation pelleted those phage particles, and so we removed any other debris. And you can see T4 and T5 still stay relatively high. The bacterial control is still lower, and the scale is very different because it was a pretty rigorous purification step. By dialysis, our final phage sample, what we end up with are T4 and T5 more enriched, and our bacterial control is almost at the level of the black. And so this is really important because if you remember, these iron 57 colloids, if they were also in the phage sample and we couldn't tell a difference between that and the bacterial control, then that means the phage doesn't have the iron 57. But in this case, you can see that iron 57 in the phage samples remains higher while the bacterial control gets really low. So this is exciting. It means we got our iron 57 in our phage and purified it so we could see the signal. So I told you we did this 3 times. This is the first time I just showed you. Another time, we again have these higher values for the cell culture, so it's homogeneous. And then we have depleting amounts of the bacterial control while the T4 and T5 stay high. Zooming in these last two, again, the T4 and T5 samples remain enriched with iron 57 while the bacterial control is pretty low. Again, the same trend, all similar at the surface, but depleting levels of that bacterial control. And then although the bacterial control is higher for this sucrose step, by the end after dialysis, those other small cellular debris are removed. So is this significant? You know, we have these different values for iron 57 for the phage versus the bacteria, but like, was it really that different? Well, it was. If I did a t-test for the T4 versus the bacterial control, T5 versus the bacterial control, and T4 versus T5, and at a significant of 0.01, a p-value greater than 0.01 had no significant difference, and lower had a significant difference. So our cell culture, our intact pinatas, all those starting bars that were similar, we actually saw no significant difference in any of the t-tests, which is good. We didn't want anything that was going to stand out as being different than the others for a starting point. Post-dialysis with my phage, both my T4 and T5 versus the bacterial control had a significant difference in value. So this is exciting. That means we're really seeing this iron 57 in these phage. And then T4 versus T5 is actually, I don't want to say surprising, but enlightening, because we didn't know if T5 would hold on to this iron. And what we see is there's actually no significant difference between T4 and T5 values. So although we don't have that protein structure for T5, it's pretty exciting that we could actually trace this iron 57 into some phage proteins in T5. So to answer my questions from the beginning, the iron 57 content per phage, I did this by doing the cyber staining of my phage samples and quantifying them, and then comparing that to the iron 57 content in that sample. And so we got a variable amount, a range anywhere between hundreds and thousands of iron atoms per phage. And you might say, like, you told me 42. Well, I didn't lie, but what I mean by that is the GP37 protein is like this little tail tip. I mean, it's a very small proportion of that entire phage, for which we don't have a lot of those other protein structures at the resolution to see iron atoms. So we never thought this was a fully exclusive number, 42. So it is exciting to see that there are perhaps other places that proteins could be complexing iron, or the capsid could actually be sticking iron. And that's been shown for other in other studies. T5, we didn't know, if it would contain iron. We found those iron sulfide binding motifs, but we didn't have that hard evidence. And so it's very exciting to see it kind of keeping ranks in some cases with the amount of iron atoms per phage in T4. So my second question was, where did the iron come from? Did it come from the bacterial host? Well, if you remember, I wanted to rinse these bacterial cells really well. And that's because I wanted to make sure there wasn't excess iron in that media that could be taken up by the phage. So the bacterial pellet went through this rinsing process, and the supernatant should have been very low in iron 57. Well, when we calculated the percent of the total iron for that resuspended bacterial cell sample, more than 93% of that iron 57 signal was from the bacterial pellet, whereas less than 7% was from the supernatant. So it would seem likely that the source of this iron 57 to the phage is actually from the bacterial cell and not from the environment, which is very exciting. So like Maya said, we published this study, this hypothesis and theory article in 2016. We kind of wanted to like mark our territory. We had the idea first. So it has kind of made a splash in these two very different fields, and it's kind of, I think, been a continued conversation about the different influences of both sides. So it's been viewed 8,000 times in the past 3 years and downloaded and even cited quite a few times. And so far, nobody's told us we're idiots. So a recent study just this year in Terra Oceans, the Terra Oceans data set was mined for these iron binding motifs that I mentioned. And what they found was throughout the global ocean, almost 90% of the viral tail proteins contained one of these iron binding motifs. And so I showed you just a couple examples of the phage. They found a lot. And then the base plate assembly proteins and the tail tip proteins also contain these iron binding motifs in different various proteins. So this is really exciting, and I think people are intrigued by the idea and obviously want to look into it farther. So just to wrap up, So we developed the Ferrojan horse hypothesis, and then we successfully developed the methods to incorporate iron 57 into progeny phages using E. coli as a model system. I was able to quantify the number of iron atoms per phage for T4 and T5 and identify the cellular source of iron to the phage progeny. So moving forward, this is all well and good, and you're like, we're in marine science, why'd you just tell me about E. coli? Well, I promise this for a purpose. These methods can actually be applied to a model system. Vibrio is a very straightforward, fast-growing model system for marine bacteria, and some strains grow faster than E. coli, which is pretty cool. So we know a lot of different phage. We have a diverse collection of those phage that we could work with. So putting these same methods into a Vibrio system would be very enlightening for the oceans, and it could get at sort of that iron cycling question, identifying if these phage could be part of the colloidal fraction for a marine phage, and if that recycling of iron is actually happening in the ocean phage progeny. So my time here at CMS has been sort of a puzzle piece, which it often is. For this opportunity, I'm so excited to see where it goes. Thank you. Water. Hey. I just have a couple acknowledgements to say, like point out. Sorry, I just said thank you to. I was just really excited. Ellie, is this part still recording? Yeah. Thanks. Okay. So a huge thank you to my committee, Maya and Kristen for being my co-advisors through the good and the bad. Karina for being a third advisor a lot of times whenever I'd come to her in crisis. Huge thank you to Salvo, our incredible postdoc who I horrified often, and I appreciate your patience and teaching me about iron chemistry and dealing with all of my rusty things in the lab. Natalie Sawaya, my statistical wizard, for all of your help and friendship. I cannot thank you enough. Mackenzie and Kima and Natalia, my lab classmates in the Breitbart Lab. You are just the biggest supporters and cheerleaders, and I really appreciate you. And Liz Fossbender, not here, but was my office mate and work wife for many years, for her guidance and support. Dana, who can't be here, but I was just here yesterday listening to this, and I really appreciate your friendship and your help. Shannon couldn't be here from the Buck Lab, and Lauren for all of her guidance in developing these methods. Betty Cutter, who provided the different phage and E. coli hosts. And then Leon Hardy, St. Pete for the modeling, Mr. George Lorton, who couldn't be here, Tony Greco for the beautiful EM images that I cherish dearly, my wonderful husband, Scott, my loving family, thank you for being here. Papa Chase, special shout out, because she deserves this as much as I do, because she was right next to me the entire time. And last night when we were practicing, a picture of E. coli went up and she started barking, and I was like, that's my fate. And Hendrix is less supportive, but and my bearded dragon Abraham, and somebody really close to me who couldn't be here today, but I think she's watching. So actually, my Nana and Papa's 41st wedding anniversary, or 41st, 61st, I'm sorry. 61st wedding anniversary, and so I think they're watching, and I love you, and thank you for being supportive and teaching me to love the ocean as well as be strong and fiery. Okay, thank you. Stop it!
All right. So with that, do you have any questions for Chelsea?
Oh gosh, I had blocked this part out. Yes.
Beautiful presentation.
Thank you.
Well done. I just had a quick question about speculation, really.
Yes, there's a lot of it.
Speculation for biomedical science, actually.
Okay.
Do you think this will have any implications for people or organisms that become anemic, that the phages are drawing iron out?
Absolutely. I could talk about this all day. And within our bodies, there's a battle going on. Not to provide more like icky analogies, but it is a war ground for iron. And so, you know, I mentioned E. coli need this because they are fighting for not just iron, but our iron that we need to, you know, to oxygenate our blood. So definitely, this could have major implications for a multitude of illnesses that require iron. A lot of pathogenic bacteria will suck up a lot of that iron, and there's a lot of complexity when you think about if you get a viral infection on top of that pathogenic bacteria, cystic fibrosis. This can be a really interesting environment. And so I absolutely think, as much as we pulled from the medical field, this could go back and translate. Absolutely.
Now, first of all, fabulous.
Thank you.
We're now atomic resolution transmission
Yes.
electron microscope. I was wondering if you thought that we would ever be able to directly visualize the iron on the tail fibers, maybe with some type of label.
The synchrotron, definitely. We've talked to some colleagues about this, and it would be a challenge because the phage particles themselves are tiny. But I definitely think that would be a way to work on this in the future. I would love to be able to just, there are the iron atoms, and look, they're all over the phage, or they're just in the tails, it's lit up. So I absolutely would love to see that happen. Brianna?
Yeah, really awesome talk. And this is probably jumping way ahead. But,
That's fine.
What kind of implications do you think this might have for the red tide Corynea-brevis trichodesmium relationship?
Interesting. I mean, I think viral infection could definitely be playing a role, and the amount of iron that's available could influence that. That's a pretty complicated system. And if there were excess nutrients, you also have the killing of bacteria. Yeah, I'm not sure. That would be cool to look at, though, and the amount of iron in a red tide bloom. which I don't actually know. But that would be pretty cool to look at. Yes.
Very nice talking again.
Thank you.
Back to the red tide.
Oh,
issue.
okay.
Well, no, the thing is, I've been sitting here thinking about, geologic history and
Yes.
the fact that the first 3/4 at least of geologic history
Mhm.
was an iron-rich world
Yes.
because of
There's no
various
oxygen.
and so then I'm thinking red tide causing hypoxia
Mhm.
and oxia, which might
Mhm.
release iron
Mhm.
to the red tide. So maybe I don't, that is,
Yeah.
I don't really have any question for you.
Yeah. No.